In still. Okay. All right, let's get going. Hello, everybody. Hello. Yay. Okay, awesome. Yeah, that's good. I'm glad there's energy. So there's more. Come sit. You got even team members. Come sit in the middle where there's nice seats there. Okay. So super delighted. Thank you, folks in Zoom land, for joining as well. We have 2 auditoriums worth of people here in Seattle, and we're just delighted to welcome you back to the 20th Merkel CELLebration. We have the woman who gave us the name celebration in the room here. Sherry, thank you guys. And it does capture that we're mixing the need to make progress with a lot of acknowledgement of what has happened so far. So this is really a wonderful event. I want to thank the people who have worked to put this together, in all the folks on this list, including especially Kate here in the front seat, who's going to be doing lots of stuff even in real time, and Karina back there. Thank you, Karina, and the whole extended team. This is a big, complicated effort. If you have a question in the room, you can raise your hand, and we're going to have a couple mics. Kate's got one, and Daniel's got one. And then online, just pop your questions into Zoom, and Kate and others will be watching. Ankita, thank you for questions in Zoom land. Or if it's not urgent, then send us an email, and we'll figure that out later. So tonight, I'm going to give you some fun overviews and highlights of what has been happening in the past months and year and such. Then Thomas Bencomo, an MD PhD student, will give us a cool science update of new technologies and telling us some of the key questions that we want to understand best, which is, why do people respond to immunotherapy and not? And therefore, how can we have the chance that that's going to happen be more likely? Geraldo's going to talk to us again about the gut microbiome, where we're doing some really interesting studies and learning a lot, and know that that's super important, and then eventually how we're going to link those in. Then Nikki and Victoria, our excellent clinical fellows this year, will tell you about clinical trials that we and others are developing that we're particularly excited about, and then it'll be time for dinner. Interspersed there are 2 amazing patient stories that kind of bring it all together. So one slide on this cancer: about 3,200 people per year in the United States will newly get this cancer. Compared to melanoma, much higher chance that it's going to come back. About 40% after optimal treatment will have it come back. Risk factors for getting it in the first place, reasonably standard for many cancers and skin cancers: older age, sun exposure, fair skin, and if your immune system isn't working perfectly, then that increases your risk. But 90% of people don't have a problem with their immune system. And then the virus that's on our normal skin, and kids have it, and it's in the daycare centers, it's on the doorknobs, called the Merkel Cell Carcinoma virus, causes 80% of these cases. We know why it's so rare. A bunch of weird, rare genetic events have to happen in order for the common virus on our normal skin to turn into this cancer. And in 20% of the cases, it's only from sunlight and not from the virus. But we've learned a lot from those 2 different mechanisms that have led us to better ways to treat it initially with surgery and radiation. And we're very excited about plans to make especially radiation be less toxic. We want more patients to get less radiation sooner. And that's going to have lower side effects and decrease the number of cancers that come back. And then, of course, what we've been very excited about for the past decade is increasing the chance that a patient will do well if they develop advanced Merkel cell carcinoma by about 10-fold. That used to be 5%. Now it's 50. So here's what it used to look like. In green, 5% of patients with chemotherapy would do well. Most would have the chemo shrink the cancer for, on average, 90 days from the first day of chemo. Then the immune system is suppressed and angry. The cancer is empowered and has learned a bunch of tricks, and it comes back and it's quite miserable. So only 5% of patients would do well, and a third wouldn't do well in the first place, even with chemo. So as of around a decade ago, immunotherapy came along increased that enormously. Lots of credit to go around for people who did basic work and allowed this cancer to be one of the very first ones to be tested for that, because it really appeared it was a very immunogenic cancer, would be seen by the immune system. And then what we want to do, and we're really on the way to doing that, is taking that good green slice from 5 to 50 to 75. You know, I'm not crazy enough to say we're going to fix everybody. We don't even know what all the problems are, but I really think this further chunk is absolutely possible, and we'll tell you a few stories about you know, how we're really getting there now. This is one of them. This matrix trial, look at all those sites there, 25 sites across the country, huge collaboration funded by the National Cancer Institute, and it enrolled in about half as much time as we thought it would, and huge enthusiasm for it. We've clearly seen some patients who were already not responding to immunotherapy now have responses when they just received their ongoing immunotherapy, which was avelumab, plus an ATR inhibitor. That seemed to do magical things for a few people, not as many as we'd like. This is a great collaboration. It enrolled really fast, and what we're aggressively planning, again, the last patient signed consent on Friday. So we are fully enrolled on that trial. It went way faster than we thought. It's awesome. What we want to do is what we've always wanted to do, which is add radiation, just a tiny bit, one little zap of radiation that 80% of patients can't even tell happened, but that will shrink the tumor and make it more visible to the immune system. So that is what we're super excited about putting in 2 weeks. That proposal goes to the National Cancer Institute, and we hope that in 2026 we'll have that sort of dream trial going forward. Now, a totally different thing. We are about to do a major refresh of our website. So 80,000 people see this website every year. It's a wonderful way to learn the basics when your medical team doesn't know how to manage this, and that unfortunately remains really common today. How did this come about? So this refresh. So this is a picture of David Groff, and in 2023, he passed away of Merkel Cell Carcinoma, and he happened to work for a company called Biolumina, which does healthcare communications and is focused in oncology, cancer. His team was devastated by losing their partner, and they decided they wanted to donate to us a major refresh and redesign, which we haven't done in 10 years, of our website. So we're deep in the process of that. We hope the revised, clearer website with lots of input from patients and others about clarity will launch in a few months. And Elise has joined us from Biolumina tonight from New York. Elise up in the back there. Thank you so much, Elise, for joining. So we're already on to now our 2 kind of really exciting celebrations for tonight. So the first one, I think pretty much everybody in this room knows, we have spent the last 4 years trying to get a major grant. It's actually a renewal, but a major grant, which is for $14.3 million, that would support 60 people at 4 institutions. We submitted hundreds of papers, hundreds of pages of documentation, and had dozens and dozens of meetings with our extended team, and it got funded on August 1. So we're now going to do something sort of fun, and Wyatt, our photographer, is going to come down here. And one of the reasons, so there's still a few empty seats here. So Mina, Mac, you guys, if folks come take the seats, all right? There's more seats. Claire, get in here. Come on. We want the room to be full because we're taking a picture this way. You guys have goodies. Okay. All right. So Max and Grandma and Denise, you can hold this in the front row. And we're going to do some fun things. Okay. So you have goodies under your seat. So grab your goodies under your seat. And now we're shifting this around. Okay. There's a little chaos here in the room for you guys in Zoom land. Okay, so yeah. Cheesy noise makers. Pom-poms. Bubbles, there go the bubbles. The bubbles. Okay, good. We're getting all fired up. We even have confetti on the screen. All right. So, okay. And then, okay. We pause for the picture. All right. All right. Everybody grab something. You guys, Vandana, grab a goodie from the back there. There's boxes of goodies in the back. So yeah, Karina, you gotta hand those out more. All right. Okay, this is very silly. Apologies. You can see things, right? Okay, good. All right. So take those doodads. Yeah, there you go. There's the streamers. Okay. And turn around and get ready for the camera. All right, here we go. Okay, is the check visible? Kind of. Wait, I got to move my computer a little bit. Thank you. Do you want the mic? We don't need the microphone. We don't need the microphone. Okay, here we go. All right, so wave. Okay. Okay. All right. Okay, so that's celebration number one. That's a big deal. So that's going to support our colleagues really around the world and at all these institutions. Now, don't put away your celebratory implements. Here's celebration number two. Becca is going to pull out prop number two. It's not really a prop, but we are celebrating Dr. Shailendra Bhatia, who's in the front row here, and he needs to stand up. And something is being ushered into the room here. Oh, I need to keep the view going here for Zoomland here. Okay, so no, wait, I can't see. They can't. Zoomland can't see that. You got to put it over here. Yeah, put it in front of Ankita. Huh? You can see? All right. Okay. All right. Well, I guess that's okay. You can't see all the people behind. Okay, we'll move it later. All right, fine. You guys tell me what to do. All right. Is it time to take that off? I guess okay, go back to the presentation for a sec, because I didn't actually say what I was gonna say. All right, so Dr. Bhatia is being named the Lynn and Daniel Lerner Endowed Chair for Merkel Cell Carcinoma. Let me tell you what that means. Okay, so these are pictures of Lynn and Daniel Lerner, perhaps separated by a couple years there in terms of the eras of their lives, but Lynn, I think had a disorder that required a bone marrow transplant, so they knew of the Fred Hutch. They're from Pennsylvania, and Daniel developed Merkel cell carcinoma and had a major battle with that, but eventually beat it. And at this point, their children have decided to make a significant gift for both graft versus host disease and bone marrow transplant in terms of the mom, but Merkel cell carcinoma in terms of the dad. And so we took care of Dan about a decade or more ago, and now we just learned about this. It's very exciting. What is an endowed share? It represents a couple $1 million roughly, and this was somewhat matched by other sources, but a big part from the family that goes into a fund that kicks out about 5% forever. And that will support, that's enough money to support a good chunk of a faculty member, such as Dr. Bhatia, who has published over 60 papers on Merkel cell carcinoma and is our lead medical oncologist and has taken care of hundreds and hundreds of Merkel patients, many of you here today. And we are delighted that he will have this for years and years. And then after, frankly, all of us are not around doing this stuff anymore, this will live on indefinitely. As far as I know, it is the first Merkel cell carcinoma focused endowed chair in the world. And it will help and integrate with our Merkel Cell Institute indefinitely to support leadership and excellent care and research into the indefinite future. Dr. Bhatia, please take your seat. Okay, I'm not sure what's going to happen here, but okay. Oh, okay, that's one thing. That was not supposed to happen. All right, this is better. Okay, woohoo! All right. So I think you need to sit. You can hold these so that they're real pretty. And I think Pennsylvania about what this means. Yeah, just talk loud. Is it on? No. It says it's on.
Maybe it's a sign.
It's on.
All right. Well, I am truly humbled and honored to be here. I did not anticipate this fully, the extent of it. I know Paul was planning something, but to make me sit here and talk, that's a little, yeah. Well, I do want to start by just thanking the amazing generosity of the Lerner family. I actually never got a chance to meet Dan, but from all I have learned about their parents, sounds like they were very accomplished people who wanted to give back to the world in so many different ways. So I really want to thank them for their generosity. And I want to it makes me actually take back in time. I came here 20 years back, and the best thing that happened to me was meeting my mentors, and Paul was one of them. And I have learned so much from him. Over the last 17, 18 years or so, while I've been working with Paul, I've gotten a lot of gray hair. And the flip side of that is Paul keeps getting younger every year. I see him. And you can see a sign of how he plans the celebration today. The child in him keeps coming out more and more every year. But I do want to take a moment to just appreciate what we are all part of together here as a community. We have patients, we have caregivers. Every day in my clinic, I'm humbled by the amazing generosity of our patients in terms of how much they trust us. You would not believe me, but there are some people in the room who will. Patients have let me put electrodes in them and generate 1,300 volts of electricity. Like, where else would you see that kind of trust?
And cure several of them, yes.
And yes, some of them have done really well, which is what kind of feeds into our craziness every year.
Yeah.
But again, I don't want to take too much time. I know we have lots of exciting scientific updates, but I just want to say thank you to all of you from the bottom of my heart. This is an amazing team, and I am just truly blessed to be part of that. So thank you.
Facing in this direction? Okay, one quickie facing in that direction. Okay.
We're going to take another picture.
Grab your pom-poms. Okay. Okay. Pom-poms. All right. 1, 2, 3. Thank you. All right. All right. Now we can Thank you. Okay, super. And we'll flip back to the presentation. That was different. Okay. All right, so a word about this institute that we have launched in 2025. We collaborate to cure Merkel Cell Carcinoma, perfect mission with a rare cancer. You can't do this by yourself. Here is that picture from Sweden just a couple weeks ago. A good chunk of the team here, and then folks from Europe and all across the US. We have 80 members across 10 countries. We run a weekly tumor board of the most tricky cases presented to us from all over the world, as you'll hear, including Serbia, leading 4 multi-site studies and 2 training programs. So the studies are a really big deal, again, collaborating with the NCI and with other sources to run these trials that no one else would be interested in or have the means to do. The 2 training programs that we're so pleased about, this one is the most important one to me, because Nikki and Victoria keep me from going completely nutso. And 300 patient reaches per week for them. It's a 1 to 2 year roll. They contribute also to our research. They get all kinds of ideas as they watch patients struggle with their challenges, and that's led to over 50 publications by 20 plus fellows over these 20 or so years, and they are 100% supported by patient donations, and that's a super unusual, magical thing. They're there to help people, whether we're seeing them, whether we're not, they're in a trial, they're not, they're just there to help. Our other program is the undergraduate research assistants, Ava, Brooke, and Mina. Over 200 people applied for these 3 slots. That is brutal. Those are 200 University of Washington undergraduates, not just random folks. So this is really a incredibly competitive thing. They contact patients. They do our PUP dates, which is patient update project, every year, where we're tracking our 2,000 patients, about 700 of which are alive and we're actively in touch with. And they did things like prepare this presentation. Thank you, guys. Then here we've got our Merkel clinical team that many of you, if you're patients, you would know well in many cases. In the red are our brand new folks: Phillips Wu, Tomoko Akaike, Catherine Jackson, Saba Kurtam, Divya Seth. I believe they are all here tonight. Thank you, guys. Raise your hand. Yep, there you go. That is awesome. We have such an integrated and collaborative team. And our philanthropy team, we would be in big trouble without them. Here is the official language. In 2022, UW Medicine and Fred Hutch consolidated fundraising at the Fred Hutch Cancer Center. That's a very complicated legal way of saying we're really partners and it's working really well. We've got the patients, we've got the research, we've got the university, they're all together. The Fred Hutch is leading the research of fundraising, and these are our wonderful colleagues, Sherry, Katie, and Megan, who are here tonight, and we appreciate their partnership so much. Thank you. This summer, we ran a super fun event. We could all do biking or walking or running. The Obliteride, we aimed for raising 50,000. We exceeded that, and that's in particular appreciation to Mike and Burma Rainbolt, who themselves donated $30,000. Thank you so much. The total donor impact is impossible to properly acknowledge. Over 5,000 separate donations over these years that have led to clinical trials that would have otherwise not happened. Blood test logistics. What do I mean by that? We've got circulating tumor DNA and we've got the AMERC antibody blood test. The latter was completely homegrown here in University of Washington. It's used around the world. Circulating tumor DNA was a big study to show that it works really well with Merkel cell carcinoma. Your donations do not pay for those blood tests. They pay for the people in our lab who make sure you can get them, that they happen at the right time, and that the results are interpreted properly and the balls do not get dropped. That is 1 to 2 full-time people, because we're running these blood tests on hundreds of people and they're complicated. So that is a really big deal. Improvements in treatments, huge, especially things like radiation, as you'll hear a little bit. We're really moving away from less aggressive radiation. It works just as well to do 1 treatment in many cases as 25. That's amazing. Many fewer side effects, et cetera. And everybody on our team is supported by donations. The little green pies show you roughly what fraction of their salaries are supported by donations, and that's a lot. Here, I want to now thank George Campbell. He was my patient in 2004, and someone else just came up to me and said, they were my patient in Boston. Thank you. That was 2005. They're here tonight. But George beat his Merkel originally in 2004, and then that wasn't enough for him. He then spent the next 20 years founding and organizing a Merkel Cell Carcinoma support group that helped over 1,000 patients. And he spent on average 6 hours a day communicating, connecting, cajoling patients into getting the right information and connecting them with people that could help with their care. He passed last year of another issue, but we wanted to acknowledge the huge impact that he has had. And in a moment, you will hear from one of the patients what that impact was. We're very fortunate that Stuart, Al, and Larry have taken over that mantle of leading this patient support group. It's continuing on, and it's very important to connect people with the resources that they need. Sometimes they're far away from centers that can directly manage their care. So we're going to do our first patient story, and we'll switch to that in a second, Daniel, but this is Jasminka's story. She is a pediatric psychologist who was the first patient, as you'll hear, in Serbia to ever receive immunotherapy for Merkel Cell Carcinoma. And she was diagnosed in the heart of the pandemic. Here she is in red on the right with two of her friends.
My name is Jasminka, and I'm 54 years old, and I'm child psychiatrist and psychotherapist and mother of two sons. And I live in Serbia. It is a small country in Europe. My city is Novi Sad, and my journey started in March 2020 when Corona started also, and I discovered a tumor mass in my groin, and I didn't have anything on my skin. And pathology reports described it is as highly proliferative neuroendocrine carcinoma. At that time, Merkel Cell Carcinoma was just mentioned as a possibility, but without enough specifiers for that. And I then underwent surgery and chemotherapy, but during the treatment with chemotherapy, tumor metastases near my kidney enlarged, and I felt devastated. I cried a lot. I felt alone, without perspective, as I was sinking into something dark and unending. I couldn't find peace. My mind was restless, constantly thinking of the worst outcomes. And then I found the MCC support group, and everything changed. And George Campbell, the founder of the group, was a true humanitarian, and he was kind and knowledgeable and generous with his time. And he helped hundreds of patients, including me. And with the guidance from the group, I contacted Dr. Astrid Blom and Mahtab Samimi from France, and they advised me to send tumor samples to Professor Jürgen Becker in Germany. And he confirmed the diagnosis of virus-positive MCC in March 2021. And that was when I finally had a name for the disease I was fighting. And it was a turning point. Finally, I had clarity. And from that moment on, I could focus on finding the right treatment. As an informed patient, I know that immunotherapy is the most effective treatment. I didn't want to have chemotherapy anymore, and I wasn't available. That therapy wasn't available in my country at that time, and I was waiting for 5 months to access immunotherapy, and it was emotionally and mentally exhausting. And I was the first patient who get avelumab in Serbia. And while I was waiting, new metastases, about seven of them in all of my body, started to grow, and in that time I had a big surgery because I was waiting. I knew that immunotherapy is the therapy of choice, but I couldn't get it. And so I finally started with avelumab, and it was I abruptly reacted. After 2 doses, all metastases were gone. So it was maybe 1.5 years ago. I developed symptoms, nausea, vomiting, and headaches, and imaging revealed a large brain metastasis about the size of an egg in cerebellum. And I underwent urgent neurosurgery, and fortunately the surgery went well, as you see. I resumed immunotherapy, and since then I have had 3 small brain metastases, about 4 to 5 millimeters, and they were treated successfully with stereotactic radiosurgery. And past maybe one year, I'm free of symptoms, even in my brain. And I would like to advise to stay informed, to ask questions, to get a second opinion. Don't be afraid to speak up, and seek out the best care. Surround yourself with people who love you, accept help, learn about the disease. The more you understand, the less powerless you'll feel. Most importantly, don't lose hope. Even in the darkest moment, light can return.
So why do some patients respond and some patients not? Thomas Bencomo is going to give us a science update. He's an MD PhD student on the team, and bringing the best technologies and the best brains to this problem. Thomas?
Thank you, Paul. So my name is Thomas Bencomo, and I'm an MD PhD student in the NEAM lab. And before I begin, I also wanted to shout out 2 other mentors that I have for this project, Manu Sethi and Evan Newell, both at Fred Hutch. They're scientists that bring sort of expertise in the new technologies I'm working with for this project. And the title of my talk is Visualizing MCC, We Need More Responders. So as Paul had shown you earlier in the talk today, we have about now, if someone receives immunotherapy, about 50% of people will experience long-term benefit, and that's this chunk of the pie in green. And while this is great, this still means that about half of patients will only receive temporary benefit, those in yellow, or no benefit at all, those in red. And so a key question we're trying to answer in the lab and with this project is, why do only some patients respond to immunotherapy? And how can we make these people in yellow and red turn into the green portion of the pie? Now, from a lot of research from a lot of different groups, we know that there's a lot of complex factors at play that cause some people to fall into the green part of the pie or the yellow or red. Today, I'm going to focus on the cancer cells or the tumor cells themselves. We know that the tumor cells are very complex, but we think there are certain features of the different tumor cells that make your tumor more or less likely to respond to immunotherapy. Unfortunately, with old technologies we've used in the past that have been the standard approach, it's been really hard to tell these features apart from one another. And we haven't really been able to distinguish tumor cells that might be better for responding to immunotherapy versus those that are kind of resistant or won't respond to immunotherapy. We have a hypothesis in the lab that is the backbone of this project, that there are 2 different types of tumor cells. We think that there are sensitive tumor cells in green, and these in the green on the left over here. We think these are tumor cells that can be seen and killed by the immune system. We also think there are another type of tumor cell called unresponsive tumor cells that can escape the killing by the immune system because they can hide from those immune cells. And then finally, just to kind of wrap up the model, we know that there's immune cells that are really important for sort of killing the tumor cells in your body, and this has been kind of known for a while. Now, in the past, when someone has a biopsy or surgery done, their tumor is taken out and looked at under the microscope by a pathologist. And it will look like these 2 images that I'm showing on the slides right here. And pathologists train for many years to be able to learn a ton of information about these tumors just from looking at these pictures alone. You can notice that there's 2 main colors on these pictures, purple and pink. And from these 2 colors alone, they can infer a ton of information about the type of tumor you have and what the best treatment plan should be. But unfortunately, even the pathologists who are very highly trained and skilled can't figure out from these pictures alone who will or will not respond to immunotherapy. It's really, really hard to predict. And so what we're going to do is use this new technology, which gives us now a 500 color view of each tumor to try to understand better why some tumors respond and some tumors don't respond. And to do this, I'm going to use this technology and zoom in on a portion of the tumor over here. This new technology, which we're calling, one way to think about it is it gives us a new gene view into the tumor, lets us look at the tumor cells like never before. So on the picture here, I've zoomed in and there's about 20 tumor cells on the slide. The first thing the technology lets us do is figure out where each cell is. So these sort of circles around the cells are the border of each tumor cell. And then the computer with the special imaging camera can take a picture of the slide and figure out the individual genes that are expressed or each cell has. And so each of these dots is a single gene that is one of those 500 colors that the computer can look at. And using these 500 genes or colors, we can figure out which identity each cell type is. So is it an immune cell or some type of tumor cell? And we can turn that picture where we didn't know what the cells were into the individual cell types. And so now we can see that there are unresponsive to the cells we think are unresponsive tumor cells in red. We can also see where we think those sensitive tumor cells are in green. And we can also see where those immune cells are, which are important for killing the tumor cells, are in blue. And so now with this new technology, we can turn the pictures that look like this that I talked about earlier, where it's really hard to figure out who will or will not respond to therapy, into a pretty picture that looks like this instead, which has a lot more information into it. So I'll walk you guys through this real quickly. In blue, each blue dot is an immune cell. And for a long time, we've known that the number of immune cells in people who respond is similar to those who don't respond. We can also see in green the cells that we think are those sensitive tumor cells, the cells that are able to be killed by the immune system. And then also in red, we can see the cells that we think are those unresponsive tumor cells that cannot be killed easily by the immune system. And we also have seen that basically the number of sensitive or unresponsive tumor cells in in people who respond or don't respond is about the same as well. And so what's really interesting is that we looked at this view across 27 different patients, including about half who responded and half who did not respond to therapy. People who respond, we asked then which patients will respond and which ones will not. People whose tumors look more like the one on the left will not respond to immunotherapy, whereas people whose tumors look more like the one on the right will respond to immunotherapy. And this is pretty clear cut based on the 27 patients we've looked at. And so with this information, the question is, where are we going next? How can we use this information? And so the first thing we're asking is, does this pattern where we have these areas of green sort of sensitive tumor cells near those blue immune cells in the responders, is this found in other Merkel cell carcinoma patients that have been treated with immunotherapy? And we're in the process of finding more patients and more patient samples to sort of test this hypothesis. And if this holds to be true in other samples, the next question is why? Why does that happen? We think that there's something about those blue immune cells that can turn the red unresponsive tumor cells into green responsive tumor cells in patients who respond, but not in the non-responders. And so we're going to try to figure out what that mechanism is and use that to make new treatments for patients that can turn those 50% of patients that don't have good long-term outcomes with immunotherapy to the patients that do have long-term outcomes. So there's lots still to work on. This is my PhD project, and I'm spending at least the next 2 years working on this. But I'm happy to answer more questions afterwards at the dinner, so please come find me if you'd like to learn more. Thanks.
My name is William Van Ice, and I'm a Merkel Cell Carcinoma patient with Dr. Neum was the Seattle Cancer Care Alliance, now at Fred Hutch. Early 2017, I was diagnosed with Merkel Cell Carcinoma. It was hard to describe how I felt because I was dumbfounded. All of a sudden, boom, I was healthy going on, and I get this diagnosis that's just almost beyond comprehension. My abdomen was full of tumors, so it's like stage 4 plus, and 5 is not exactly, it's terminal. So they both said I'm close to death, and they were very surprised I lived. Woohoo, I like that. I got this email from Fred Hutch about fundraiser for climbing the stairs in the space deal, 632 stairs. So I formed a team, and I'm wearing a T-shirt that and the name of my team was Never, Never, Ever Give Up. On the back it says Be a Warrior. So I raised, I don't know, $1,000 or something like that for Fred Hutch, and then a good friend of mine, Ed Hirschberg, and I did the top donut wave, so we got a donut when we got up there. But that was like 19 months after being diagnosed, and I could climb the space deal. And so I'd say that was a pretty good accomplishment. There is a test a DNA Merkel cell carcinoma test that tests for the presence of the Merkel cell. April 2024 was 0. Well, the next test was in December 2024 and it went up. And then the next test somewhere in March went more up. So I'm back on Keytruda. And so two infusions of Keytruda, the cancer went down to nearly 0. Now that was pretty absolutely amazing. So you'd think like this was a storybook ending, like, you know, and lived happily ever after. Not. And as Paul Harvey would say, now this is the rest of the story. This Keytruda started my kidney start failing. And it wasn't caught until sometime in 2025, in, I don't know, May, June timeframe. At that point, I stopped taking Keytruda. But however, my kidneys started to fail. And this was actually so much worse than the cancer. I got released from the hospital in April. And so it was after that the cancer came back. Well, that's not the story you want to hear. So now what? This was a very rock and hard spot situation. The Keytruda killed the cancer, but also caused nephritis in my kidneys. So after that, stopped Keytruda, and my kidney functions have gone back up. Well, that's where it stands today. So right now, according to the tests, there's no detectable cancer, and my kidney functions are back. So what's next? Well, let's wait and see. Who knows what's going to happen? But I have very much confidence that whatever it is, it's going to be okay. And as far as support is concerned, well, it was already built in, family and friends. And they gave me a lot of support during the cancer, and especially during the nephritis, because I was so sick. So for those guys who are out there, you know, facing Merkel cell, I think that, you know, it's how you approach it is so important. First of all, friends, family, being there, part of your life, giving to you, you know, support and confidence, and just believe that it isn't going to be the end. I say, you know, I didn't focus in on the sorrowness, the sadness. I focused in on life and live a life every day. I lived through all that. So the lesson is never, never, ever give up. That's my motto. And that's how I live my life. And I'd like to acknowledge my wife, Sandra Van Ice. She was there for me the whole time, you know, through the pain, watched my meds, took me to medical appointments, and I couldn't have done it without her. Thank you, Sandra. And so as far as resources, I don't think I could ask for anything more than I had. Friends, family, and never, never, never give up. Thank you.
Awesome, William. Thank you so much. Okay, good. So Geraldo Rodriguez is a super talented graduate student about to defend his thesis next month. Going to do a brilliant job describing B-cell biology, but now he's already kind of gotten a new grant to support a next chapter in how the immune system talks to the gut, which he plans to be co-leading as a postdoctoral fellow on our team together with Dr. Megan Koch. So.
Hello, everyone. I think Paul already gave up the ending, but yes, we're going to start doing this new research with a fantastic immunologist who's somewhere here in the audience. Megan, if you could say hi, there she is. She's she's joining Team Merkel. We're not joining Team Poop. I think it's the other way around. But in any case, so thank you all for coming. Thank you, Paul, for the introduction. And as Paul mentioned today, I want to talk to you about the gut microbiome. So these are essentially tiny microbes that live in your intestines and that are super important for regulating your nutrition as well as your immune system. And we think that by learning more about how the microbiome works in everyone, we could use what we learned from them to either improve current immunotherapy treatments for Merkel cell carcinoma, or even come up with totally new treatments that will complement the current technologies that we have. So going a little deeper into what the microbiome is. So the gut microbiome is like these bacteria that are in the trillions that live in your intestines, essentially, or your gut. And you can see them here in this diagram. You can see a few of them that are essentially within your intestine and are surrounded by your intestinal wall. And their key function, or one of the most important functions that they have, is that they help you digest food that you would otherwise not be able to digest on your own. Now, once you eat that food, these bacteria eat that food for you, and then they create these essential nutrients or metabolites that essentially keep you healthy and keep your immune system functioning to essentially get rid of infections and potentially fight cancer. So now you could imagine that because you have trillions of these little microbes, your body needs to find a way to regulate how those microbes live within your intestines. And in a healthy gut, what you want is that you want to keep that balance of bacteria within your intestine at a level that will continue digestion in a healthy way, but not in a way that you either deplete them, because again, they don't belong like to your cells, but also you don't want to overpopulate them. So I'll give you an example of what that could look like. So you could either have bacterial overgrowth if your body doesn't control these bacteria well enough, or you could have bacteria, as shown here at the bottom, or you could have bacterial depletion on the top if your body's like, there's too much of this. And both of those things are actually associated with something that we call a leaky gut, which is essentially a tear in your intestines. And that tear itself has been associated with a lot of really bad things, including inflammation and cancer. So as you could imagine, there's this very important need for your body to keep these microbes at bay. Now, how does your body do that? Well, there's essentially two big things. And one of them you can actually start fixing or improving today: diet and your immune system. Very big things that regulate the microbes living in your intestines. Now, how does diet control the microbiome? Well, so I'm giving you here a more in-depth picture of what your gut looks like. Again, you have your intestinal wall. There's these little cells that are called epithelial cells. And then you have your microbes living in this like yellowish mucus. So essentially it's a goo that your intestine secretes, and it sort of creates like this layer where the bacteria live. Now, good bacteria require fiber to not only promote that growth of mucus that keeps a little cushion that protects your intestines, but it also helps prevent bad bacteria from colonizing your intestines. So fiber is the key thing here that essentially allows you to keep this nice little cushion layer that protects your intestine and allows the good bacteria to keep growing and preventing bad things from happening. On the flip side of the coin, if you have a high-fat diet with very low fiber, then you sort of start depleting that mucus layer that requires fiber to grow. And then on top of that, the bad bacteria really like fat. So they start overpowering the good bacteria. And essentially, you have two things that happen here. Number one, you deplete that mucus layer that allows the intestines to stay healthy. And number two, you allow the bad bacteria to then start tearing apart your intestines and create these like tears. And that's what the leaky gut is. Now, on top, this, of course, is something that we can mediate with diet, but our body also developed a way to essentially mediate how your bacteria are regulated. And there's a lot here that we don't know yet. You can ask me, and you can ask Megan in the audience later about how exactly we're investigating these mechanisms to better understand how we can mediate this to improve cancer outcomes. But roughly speaking, you have your bacteria living in your intestines. There's another layer of complication where you have these bacterial sensing cells. They're immune cells that can essentially probe which bacteria are living in your gut, grab them and say, okay, this is good or this is bad, there's a problem here. And if they detect the problem, they can actually recognize the bacteria that are bad. And they send a signal to these so-called antibody secreting cells, which are actually B cells. So, going back to what I do, but essentially these antibody secreting cells, as the name says, can release these antibodies that are specific for the problematic bacteria. Then that antibody can be released out into where the bacteria are, and they can essentially stick to the bad bacteria and then send more signals to bring down that bad bacteria from becoming worse. Again, there's a lot of things that we don't know here. I'm sure that Megan will be like, what are you talking about? Like, this is not completely accurate. So my apologies, but that's why I think there's a lot more that we could do and we need your help for this. There are two things that we are gonna request your help for. The first thing we wanna say is, you can actually change some of this. We can promote a healthy gut by, again, having fiber in your diet. And there's very different kinds of fibers. But today, as of now, if you're not on diets that include a lot of different fibers, I strongly recommend you do. Here are some examples of the foods that you could go and start eating and enjoying, because bananas and apples are great. Come on. Paul doesn't like bananas, by the way, so he's a little weird in that. But there's potatoes. He likes potatoes. So again, starting today, nutrition with fiber, super key to keep a good, healthy microbiome. And the other thing that you can help us with is, and this is what Paul essentially ruined at the beginning, you can help us by donating your stool and blood samples. So again, there are a lot of unknown questions. And this is true not just for Merkel, but for melanoma and many other solid cancers. Lots of things we don't know. And we really need our community to come together and essentially answer all these questions that we have. We recently were awarded $150,000. Again, Paul just gave it up at the beginning to assess the gut microbiome and immunity in MCC patients. And this is in collaboration with Dr. Megan Koch, again in the back. This was going to be her formal introduction to the Merkel team. And if you're interested in participating, please email us and let us know. But yeah, so two things, diet and poop, the new gold rush. So with that said, I would like to thank everyone here today, the DM Lab, all our collaborators, Megan and Madeline, who's a grad student in her lab, who's going to be helped. There's it, it's her. And all of you for your amazing contributions, for being here today. This is truly an honor, and for being silly with us, because as Paul is becoming younger. Thank you all so much.
At least sillier. We'll see about the younger part. All right. Okay. Great. So then now we're our last little bit is a very important one. And it's about clinical trials. And again, Nikki and Victoria, please come up. Am I missing something? No. Okay. When there's talk in the front row, I'm worried there's something going wrong. Anyway, thank you guys for all you're doing for our patients and for giving an overview of what options are for their care.
Hey, hello everyone. I'm Nikki. This is Victoria, and as Dr. Nia mentioned, we're his current clinical fellows this year, and we are going to be doing a brief update on clinical trials for Merkel cell.
As of today, there are 47 active trials in the US that include Merkel cell cancer. Although only a few are designed specifically for Merkel, today we will be discussing the trials that our team is involved in and the ones outside of our team that we are excited about. Here's a map showing a lot of active clinical trials in the US that you can see as dots on here.
Yeah, and we split up our talk today into 3 short topics about clinical trials. We're going to start by talking about trials that work towards preventing Merkel cell from recurring after being treated, then move on to treating Merkel cell that is resistant to standard therapies, and then finish by talking about how patients can find clinical trials that would be appropriate for them. So we're going to start by talking about trials that look at preventing Merkel cell from recurring. And we use the term adjuvant to talk about therapy that started after initial management with surgery and or radiation. So to take an example of a patient who was treated initially with surgery and radiation, and if they have no signs of cancer that we can find, but are high risk for the cancer coming back, they can enroll in a clinical trial looking at adjuvant therapy. So they would be randomized into either being monitored only, so observation only, or they can start immunotherapy, which is what we call adjuvant immunotherapy. And there are trials all over the world researching different adjuvant immunotherapies. Of note, there's a trial in Germany that just came out with results. They were not statistically significant, maybe showed some preventive benefit, but not super conclusive at this point. So the other trials, 2 in the US and at least 1 in Australia, we are waiting for results to come back on those. And we're hoping that the data from these trials can help guide our recommendations on adjuvant immunotherapy moving forward. But since we don't have these data right now, we do not routinely recommend giving immune therapy to patients if there is no evidence of Merkel cell after initial management.
So what if a blood test like the ctDNA test tells us cancer is coming back, but we can't find it on their scans and on their skin? Over 80% of these patients will have their disease recur within a year. One of our doctors, Dr. Tomoko Akaike, wants to run a trial through the National Cancer Institute that will test if immunotherapy is beneficial to these patients. Now I'll be touching on our next topic, which is treating resistant Merkel cell cancer. So what do we mean by resistant disease? When a patient has advanced Merkel, they usually receive immunotherapy, which is often Keytruda or pembrolizumab. About half of these patients will have ongoing benefit of this treatment and see their cancer reduced without further progression. Unfortunately, about half of the patients will either not respond to this treatment or will have their cancer eventually progress while on it. There are numerous clinical trials studying what the best next treatment is for patients who need another option.
And Dr. Neum mentioned this, but the Matrix trial, very exciting to say that the enrollment is now complete. We are fully enrolled for that in record time. And there were some encouraging results with the new ATR inhibitor regimen, but most patients did not necessarily benefit from the ATR inhibitor regimen. So we are very excited about the possibility of adding in radiation to the mix with the Matrix 2 trial, hopefully coming soon. But we wanted to give you an example of someone who benefited from the Matrix trial, and one of our patients kindly gave us permission to share his story. So this is a patient who had Merkel cell on his right cheek with involvement of his right neck lymph nodes. And he was initially treated with surgery, radiation, and immunotherapy, but unfortunately his disease came back. So he was then treated with another round of surgery and a combination immunotherapy, Ipi-Nivo, but his disease continued to progress and grow. So he moved and transitioned to another immunotherapy, Keytruda. And again, his disease continued to grow and spread, just very stubborn. So he started on the Matrix trial and had a complete response, which means we cannot find any evidence of disease in him at this point. So very exciting. Yes. We wish this kind of response happened more often, so we are very excited about the possibility of adding radiation in and coming up with new treatment regimens to hopefully have this kind of response more often.
Okay. TRIC MCC is one of our homegrown trials run by Dr. Miller and Dr. Bhatia. It works using a combination of 3 different therapies to remove the brakes off of immune cells to help them attack cancer easier. Enrollment for this trial is complete, and we have seen some positive responses to the therapy. This will be guiding next steps, such as possibly trying other therapy combinations.
All right. And lastly, we are talking about tarlatamab, which is a new type of immunotherapy. It essentially acts as a glue that connects your immune cells with the cancer cells so that your immune cells can better break down and kill the cancer cells. And there are 2 current trials right now, one at UCLA and one at the University of Arizona, that are looking at this therapy. And we're excited about this because it could be a promising option for patients who are eligible and have resistant disease to our standard therapies.
Okay. Next, we will be talking about how to find a clinical trial specific to you. So to learn more about clinical trial options that could work for you, we encourage you to reach out to me and Nikki. The best way to reach us is through our email here at mccteam@uw.edu. Another option is to directly find trials through clinicaltrials.gov, but this website can be tricky and a little bit difficult to navigate. Lastly, AI is a useful tool. So here I asked ChatGPT, I am a patient with Merkel cell cancer. My cancer is not responding to pembrolizumab and I want to enroll in a clinical trial. Which ones can I enroll in? And it gave me a wonderful table of trials to look into. So I thought, great. Next, I got more specific and I tried from the point of view of someone living in Tennessee. So I said the same thing, but I said, I want to enroll in a clinical trial close to me in Tennessee. ChatGPT showed me 2 open Merkel trials in Tennessee, showed me one that's a little bit outside of Tennessee, and it showed me some cancer centers that would be good resources. So overall, it actually did a really good job searching for possible leads. And this is a very good starting point for discovering options close to you for clinical trials. This QR code goes to our monthly newsletter, which summarizes trials and important new findings. It's an ongoing set of updates similar to what we presented today, and it's a great resource. So if you are interested, I encourage you to scan the QR code here. Thank you so much for your time today in learning about recent clinical trials. Please don't hesitate to reach out to me or Nikki through our email if you have any questions or if you want to learn more. Thank you.
Yay. And that's it. So it's time for dinner. We are 3 minutes late, not too bad. Everybody on Team Merkel, that includes folks from the other auditorium, please come down. We will take our annual Team Merkel photo. Everybody else can go get a head start on the food. Thank you so much for coming. We couldn't do it without a big team. Bravo.